Fabio, as usual, he finds something extra to do, either on the phone or some meeting, sudden meeting that he has to go to. So I'm here to introduce our first speaker, Elisabetta Balzi. So if you were here in the morning, you've heard the extended description of the profile. I will give you a shorter one. She's currently an active senior advisor, but she was the former head of Ocean, Seas and Waters Unit and Deputy Director of Healthy Planet at the European Commission, DG Research and Innovation. Before that, she was head of Bioeconomy Unit at the European Commission, Joint Research Center. She's in charge of the EU research innovation on marine inland waters, as well as the EU missions to restore our oceans and waters. She has been in charge of coordinating the EU framework program design, implementation, and international collaboration on food, agriculture, forestry, marine, biochemistry, biotechnology, and bioeconomy. From FP6, which is more than 10 years ago, 15, up to Horizon Europe. So you understand that she has tremendous experience on funding and on what is going on. And with that, I would like to ask her to come up and give the first presentation in this EU cluster on bioremediation, which is a very interesting initiative, supported generally by EU, that loves to do clusters as well.
Good afternoon, and good afternoon again to all of you. I think we already saw each other, most of us, this morning, so I will not, of course, repeat my presentation. I'm very pleased to be in a project meeting. We don't often have opportunity and the privilege of being very close to the clusters. It's very important indeed to be here. It's very important that you are together, that you are discussing, that you're not working within just your consortia, which are already very big projects, of course, but that you are really making a community of projects which cut across. I've seen cut across also different programs, because some of you belong, most of you belong to the cluster 6 that I presented this morning, but I see also there are some Marie Curie Sklodowska action projects. So it's good that you keep, this spirit of community and that you work together. I will be more on the listening mode. You are on a very important subject, which needs to be put, of course, in the current policy context that I described to some extent this morning as far as water and ocean are concerned, but I know you're very involved also in beyond the ocean, but also on land. So it's very important that you take into account the new political guidelines, that your projects are, providing impact and in the current context. I mentioned this morning, the policies, the new Ocean Pact, the new water resilience strategy. I encourage you all to have a look to these main documents. A new Mediterranean Pact will be upcoming, but I also mentioned that there is a new vision for agriculture and food, a clean industrial deal, the Circular Economy Act, and life science strategy. All these documents are very important. So I really recommend that you, within your activities, within your clusters, have a look and align to the current policies and how we have to deliver, including on the issue of competitiveness, for example, and sustaining our quality of life, food security, water and nature, which are key principles in the new EU political guidelines. So this is important. Impact, it's very important, and that you are working all together across the program, across the EU programs also. I mentioned this morning that we also have tools which allow you and us to identify very quickly a cluster of projects on the same area, but across 16 EU programs, including the structural funds, including other EU funds. And it's also very important that you keep, you try to keep a broad picture and join forces, because we have really very ambitious targets ahead, and we need to work all together. In terms of, I know you're mentioning the title of my presentation here, but as I said, to a large extent, I referred to it this morning also. What is the situation of our policies? And this I wanted to say, the main message is have a look to the new policies and try to align and see how you can deliver with impact. How can you help us also to monitor the impact of your projects, that they don't remain, within your consortium or even within your cluster, but they can really reach out. I mentioned this morning the missions, and some of you are also mission projects. I focused on the mission Ocean and Waters, but of course, mission Climate and mission Soil are very important for you as well. You have, with microbiomes, with all the bioremediation, you can have really a very powerful impact for those missions, and the purpose of a mission is to bring the solutions to the territory. So within the remits of your project, I also really would like to recommend that you make the link to facilitate this reaching out to those national, regional, local authorities who need your solutions, to link also to the projects who might have more this focus now on demonstrating the solutions and facilitating the deployment, which are mainly in the mission, but not only in the missions. So also this is a recommendation that I would like to give, even though I'm looking more forward to listening to you. But I think this is very important to keep the broad picture, to know that we need to deliver those solutions which are stemming from your projects. They need to be, concretely helping those who need them locally. So in whatever way you can do it within the remit of your project, or linking to those other projects which have this purpose. In terms of funding, I think we will share these slides which have the details of topics portfolio that we are supporting in Cluster 6. I presented the portfolio of also Mission Ocean and Waters, but of course there is an equally important portfolio in Mission Soil, Mission Climate, which are relevant to you. I invite you to have a look to that. All this is publicly available. What is important is also that we have the current call open, 2025 work program, open until September, you probably know, and there are many opportunities also, including in the areas of bioremediation. I showed some this morning under the destination of zero pollution of our cluster, and also under the mission. So I encourage you to have a look to those programs, but also now, as we said this morning, for the first time we also pre-published very much in advance what will be the upcoming Work Program 26 and 27, which is also public. It is in my slides, and there you will find also an overview of what are the draft proposed topics at this moment. Of course, there is still a process ongoing, so there can be changes, there will be changes, but that gives also an overview of what is in the pipeline, and I would invite you to have a look to that, to keep this prospect. So very good that you gather together. We have done this, for example, when we did the mission forum for our Mission Ocean and Waters in Brussels in March. We called, at least we invited all the projects, not only of our mission, but also the ones related to objective of a mission, but which belong to the other parts of Horizon Europe. We also invited projects from other programs, in our case, LIFE or MFAF, so that we really make this community broad, and in order to join forces and really work all together, because as I said, the objectives are very ambitious. There is an urgency to act and to bring the solution. So that, I think, would be my main message. For the rest, for the details of the topics, I have here a quite detailed presentation, even with the list of what is public. I will not run through this. I think it would be too detailed, but I'm looking forward more to listening to you. But this is available, and we will make this available, this presentation, and you can browse through all the possibility. But What I really would encourage is that you keep the spirit of clustering, grow as a cluster, spread out. I'm sure with the agencies who are following you, this can also be arranged. It's also done in the frame, as I said, of a fora that we organize. It's very important that we join forces, to reach these ambitious objectives. What I also wanted to recommend is if you're working on, in one way or the other, on waters, or waters, seas and ocean, please adhere to the Charter of Emission. The Charter of Emission is a frame where we gather all those who act for our mission, not necessarily the projects which belong to the mission work program, all other projects which in one way or the other, I'm sure yours do as well, as long as you deal with waters or ocean and seas, please adhere in whatever program you belong, because there we are also gathering our communities. And you will see we have more than 1,000 actions now in the mission worth 8 billion euros. Beyond what we invest in our mission ocean, up to now we invested €500 million, but there are many actions which go beyond our own work program, and there also it's very important to have all the communities together, and then we facilitate, once you belong to the community, we also facilitate the exchanges. For example, in Brussels we invited all the projects which, belonging or not to the mission work program, they were adhering to the charter, and adhering to the charter is very simple, nothing to do with a submission of proposal that you were successfully going through, but it's just a questionnaire, very short, a form where you describe your action and along the objectives of a mission very briefly, and then you get a confirmation from us that you belong to the charter. It doesn't commit financially, neither legally, but it's a very important step to belong to it. And then we facilitate, because we have all sorts of actions to link among the different communities that we have, including to the communities of those who have to implement, because we work closely with regional authorities, local authorities, so there you can also find the link to what I said in the beginning, which is very important for us to bring the solutions that you're developing to the territories and to the realities. So through this mission charter, and I will provide, maybe I don't have it now on the slides, but it's very easy to find if you just Google mission charter, ocean and waters, and adhere to that, because that helps really to work together, including with the authorities that we are gathering there and who are really looking forward to benefit from the solutions that you are developing in your projects. So to make your project really reaching out and benefit the territories in their own context. These are my main messages. I would leave the slides with all the details. There is a very long list of topics, but I think this is beyond the scope, and I would like to listen to your presentations. Thank you very much.
Thank you very much. I should say that we will put the slides that Dr. Balgi presented in the morning on the website, so you could go there and see different calls and how much money has been spent, what is the future, etc., which is going to be quite important. We will go with no question periods because we are tight on schedule, and we will start with the series of presentations, correct?
Yes, we will start with all EU projects ongoing that will be presented for less than 5 minutes. So, and we start with NYMPHE and Giulio Zanaroli from the University of Bologna.
Thank you, Philippe, and keeping the time will be the main challenge for the project. So NYMPHE has started 2.5 years ago. It was funded under the topic Zero Pollution 110. and actually it's a collaborative project involving 18 partners from 12 countries. We are focusing specifically on 4 real contaminated sites that are characterized by the presence of mixed contaminations, including aporaic zone, municipal wastewater, industrial and agricultural soils, which contains different types of pollutants. To make it very short, we are trying to address this complexity by developing bioremediation systems that are based on assemblies of different biologics that are first selected because of good degradation performances alone, and then are assembled in the real contaminated matrix, first at laboratory scale and then at pilot scale, where of course they are also assessed from many points of view. Just to show briefly what we are doing now regarding municipal wastewater, we are already testing some technologies at pilot scale. For example, let's say assemblies of adapted population of electroactive bacteria in the Metland, which are turning very effective in the removal of a large number of pharmaceuticals, but still not full removal, which is further complemented and refined by sorption process using clamshells. With municipal wastewater, we are also exploiting microbial assemblies that have been selected on different pharmaceuticals, which are applied in a pilot scale MBR, so far actually showing very nice average removal rates for many of the target pharmaceuticals. Moving to industrial soils, here we are starting, let's say, pilot test this summer. We will actually target the surface soil through rhizo-remediation by assembling both sunflower or tobacco plants with bacteria with plant growth promoting activity and hydrocarbons degradation activity. Here you see, let's say, some examples on laboratory test, which showed actually an improvement in the plant growth, but also in the removal of some hydrocarbon classes using this assembly. We are also addressing the lower soil through, let's say, the application of sequential assembly of microbial communities in biostimulation and augmentation tests in situ. Regarding the agricultural soil, the main technology is ecopiles to address plastics and pesticides biodegradation, both exploiting assemblies of microbial communities that have been selected, and you see on the bottom left some results on plastics, but also exploiting different earthworm ecotypes that are applied for vermi-remediation into this kind of, let's say, bioremediation approach. Finally, regarding the hyporheic zone, some preliminary test in situ has been already performed, again applying microbial communities that are able to degrade target pharmaceuticals under oxic and anoxic conditions. And here you see, let's say, the experimental setup with the main actual tests that are again starting this summer or are in progress. To conclude, I must say that this is only a small part of the project. An important aspect that we are also trying to address is the development of platforms to improve the performances of these microbial biologics, which include very different approaches. Just to mention one, also, let's say, the modeling of microbiomes and the design of modulation strategies to improve their performances. And this is just one example done on the industrial soil, where using this approach a number of, let's say, chemicals that could be used for biostimulation have been identified using these models. Actually, we are in the phase of testing them to validate the approach. With this, I have to conclude. There will be, for sure, much more to come in the next months, and hope to provide you new updates in a while. Thank you.
So the next project would be presented by Thomas Reichenauer. It's about project MIBIREM.
Yeah, thank you, Philippe, for the introduction. Yes, I will talk about, will shortly introduce the project MIBIREM to you. So MIBIREM is similar than the previous project in the slightly above the middle of our time, and we will end in March 27. We have 9 partners altogether. Sorry. And this slide shows a graph, what we are doing in MIBIREM. So we are focusing on microbial degradation, not only by individual strains, but also by microbiomes. And the aim is to develop tools that can be applied also to other contaminants than we are working on in MIBIREM. So we started with sampling several sites across Europe and doing enrichment cultures with the aim to cultivate and improve these active microbiomes, but also to better understand the degradation pathways of the contaminants we are working with. And then we go on to prototypes with the aim to adapt these microbiomes to pilot tests. In addition, we are doing a modeling to better predict bioremediation at sites. And an important part is also to conserve these microbiomes and also individual strains via cryopreservation to provide them for other projects in the future and also for practical application. So these are some of the tools we are working on. As I said, the cryopreservation of microbiomes is something new. We need some quality criteria for that to develop. We work also on the development of qPCRs to better monitor the bioremediation in the future. And we adapt existing methods for single strains to degrade microbiomes. So the aim is here to improve the microbiomes by advanced laboratory evolution. And then, of course, we do mesocosm tests to finally end up with the pilot. We are working on 3 contaminants. These are HCH, not only lindane, so not only the gamma isomer, but also the other isomers, on petroleum hydrocarbons and on cyanides. And we sampled 13 sites across Europe, and now we are planning, at 4 of these sites, we are planning currently the pilot, and this will start in this fall. We have done all our sampling and also the generation of microbiomes. There are more than these, but these were the ones that were selected for isolation of individual strains. We have isolated these, and currently we started whole genome sequencing of these strains. These numbers are not absolutely up to date, so the work is still going on. The aim is to have the whole genome sequence for 10 strains for each contaminant. Yeah, this was my short overview. Thank you very much.
Now we move to the next project, BIOSYSMO, that will be presented by Sarah Gil Guerrero from EDENA.
Okay, so good afternoon, everyone. So BIOSYSMO project is devoted to the development of bioremediation systems by exploiting synergies for the improved removal of mixed pollutants, to have more clear image of the project in numbers, we are a 48 months initiative and we are currently ending our second reporting period. Therefore, we are more or less 1 year ahead from the end of the project. This project involves 13 partners from 8 countries across Europe and was expected to start in a innovation in a TRL from 2 to 3, and hopefully we will achieve TRL 4 to 5 by the end of next year. So, although I will try to focus more in the key results, let me give you a brief understanding about the objectives of the project. So, within BIOSYSMO, we want to optimize existing and develop novel synergetic biosystems, to expose them to a wide range of contaminants, therefore gaining vast knowledge in the different type of interactions, to support these developments with computational tools, and also to evaluate the best performing systems in field studies. All of these obviously evaluated within a multifaceted analysis about the impact of the technologies, considering life cycle and the environmental risk. So, BIOSYSMO is traced as a streamlined pipeline that will allow us to accomplish all these objectives. And in the first part of the period of the project, we focus in grounding the innovations. Therefore, we took advantage of the 14 sites that are part of the BIOSYSMO project to exploit the microorganisms that has pollutant degradation potential, and we both screen those diversity and enrich and isolate the key players. All of this supported with the computational tools that allow us to identify metabolic traits and interaction patterns, and later include them into models that will support the design of the biosystems. In this case, these biosystems are targeted not only against a specific pollution, but also specific remediation techniques. We have 5 case studies, each one devoted to a specific remediation technique, and I'm going to present briefly some of the key results from each of these case studies. So in the context of, let's focus in soil and sediment at the beginning. So for sediment phytoremediation, we have, well, the partners from THEME are already presented today. They have accomplished the development of consortia, bacterial consortia, able to degrade both pharmaceuticals and to achieve metal removal. And not only that, but also the development of an enhanced approach that will allow the attachment of these microaggregate consortia in the rhizosphere of the plants for phytoremediation. In the context of soil phytoremediation, the colleagues from the University of Marie and Louis Pasteur, together with the Polytechnic of Madrid, they have not only identified new fungus that has a strong metal resistance, but also they have combined them together with bacteria, so they improve the synergetic effect with the plant regarding the plant growth promoting, and also enhance the metal tolerance, all combined with a strategy in which the poplar plants are modified so that it is increased these capabilities. Regarding soil bioaugmentation and biostimulation, colleagues from the University of Burgos, in collaboration with Joseph Stefan, has advanced in the improvement of consortia able to degrade PAHs, lindane and anthracene. And in addition, the colleagues from the Imperial College of London have worked in the integration of engineered pathways for lindane in some GMOs. All of this has led to a strong collaboration with the project MIBIREM, and in particular with Simona from the University of Pisa, and we are getting really good results. And in the scope of water treatment, we have 2 approaches, the water phytoremediation, in which as key results we have been able, again the University of Burgos, able to optimize the consortia for PGPR activities from the rhizospheric soil inoculation, and they have demonstrated the performance in combination with aquatic plants for the removal of metalloids. And finally, in the field, in the targeting bioelectrochemical systems for the treatment of groundwater. The colleagues from LATAT has advanced in two main designs, one related with the treatment of metals, another with hydrocarbons, and for both they have been advanced. In the first case, they have enhanced a two-electrode design that allow a stable electroactive biofilm formation, and in the context of the hydrocarbon, they have made some advances in liquid cathode design that allows the hydrocarbon degradation and cuts the energy use, and it's also a good approach because it will enable the implementation directly in an aquifer and not need for pump and treat pretreatment. And other key results that I would like to highlight, so BIOSYSMO has a strong computational core, and in this scope, from ISLANDR, we have developed new protocols that will allow the 16F functional inference and combines proteomics and hidden Markov models. We have also developed a metagenomic pipeline that we will try to publish soon, in which that allow us to support all types of metagenomic data. And finally, and I think that's something that will be worthy to mention, is that we have generated databases that integrates most of the resources that are commonly used, such as IAWAG, MetaThic, KEGG, all in one, and we have already uploaded them in Zenodo, so they are there for your use. And finally, other key results are the advancement from the Joseph Stefan Institute in the context of enrichment and isolated approaches, and from the CNRS in the development of analytical methods for multi-residue analysis, which is key to understand the actual degradation. And yeah, from now to in a year, we will try to take all of these key results to field. Most of the technologies will be tested in mesocosm, but it is worthy to highlight that both the bioelectrochemical system for hydrocarbons and the poplar, the phytoremediation in soil using poplar, will be taken both to pilot and field study. And yeah, that's all from our side. Just don't hesitate to contact both me and my colleague Lila, that will be in the panel discussion for any further information.
Thank you, Sarah. Now we move to SYMBIOREM project, and this is Lair Ruiz-Rubio that will give the talk. She's from the University of Basque Country.
Okay, thank you very much. So, I am from SYMBIOREM, or the other sister project of this call. So mainly our target is the same of the previous one. So we are a consortium all around Europe, 5 team partners. Also we have a collaboration with US, with the University of Fresno, California. And our aim, as all of us, is try to clean a little bit Europe, let's say. So to do that, our focus activities are on soil, surface water, groundwater and sediment, and also seawater and marine sediment. So we have novel bio-based technologies that we are going to test in 12 different locations all around Europe, mainly focusing in Spain, UK, Austria, Poland, and Sweden. And also, since we have a collaboration with US, we have also other study going on in Central Valley in California. So, focusing or beginning with the strategies that we have been developed in soils, let's see. Until now, we have successfully isolated, identified, and assessed carbon-degrading autochthonous strains from several contaminated soils that I had been presented before. We have developed a biostimulation strategy with microencapsulation of nutrients first at mesocosmos scale, later at semi-pilot scale. Then also we have a bioaugmentation strategy, selecting microbial consortium that had been inserted, let's say, in a polysaccharide-based carriers. Also, we have safely carry on a gene bioaugmentation strategy. To do that, we have modified Pseudomonas aeruginosa, sorry for the pronunciation of that, with autotrophy to methionine, and they are able to degrade toluene, and all of them have been done by using these genetic engineering strategies. Also, moving to the phytoremediation, we have been developed a new technology of phytoextraction of metals. If you had been the chance to yesterday listen our colleague Christophe talk, he were explaining about our water-absorbing geocomposite and their role in this improve of phytoextraction of metal from contaminated soils. Also, we have developed a bioelectrification enhanced phytoremediation of pH, and also quite successful for hydrocarbon contaminated soils. I didn't put there, but also the strategy works quite good in metals like lead or arsenic. And also from our colleagues from US, we have a phyto management strategy in which we are using saline drainage water to grow guayule, that is a plant who tolerance salt, who tolerance boron, and from that plant we also are able to obtain natural latex and resin as latex to do gloves and those kind of things. So it's quite interesting for the future. Moving to water and sediment strategies, we have designed a cascade bioreactor from which we are not only able to remediate pHs, we also are able to recover some interesting metals. Also, we have developed immersed active surface surfaces using bacteria, yeast, and fungi that we are right now combining with a floating island. Also, some natural-based bioremediation solutions based on the combination of strategies with living mussels. So we use mussels first in lab scale, and now, as you see, a ver si soy capaz. As you see here, we have a mesocosmos scale in which we are testing how is the performance of these mussels species to clean up the water. And finally, we have natural-based solution for cleaning up the landfill leachates. So this is the approach that we had been carrying on until now, and now we are finishing this part and we are scaling up. So first, we are scaling up quite big. I am Basque. I love the things going big. So we are scaling up in Osobowice, Poland, field scale tests with the technology that we have developed, and in the Basque Country in Spain, we are going to perform small biopies like 12 tons or so. Moving to the water strategies, we have now two different setups of our wet floating islands, one in Thames, in Lake Gallion, that is close to London, another one in Tolpa Park, in the center of workflow in Poland. If you are able to see it, it's already there. And we like a lot to try to speak up about our technologies, try to convince the people how necessary are those activities. So we have a trust building and citizen engagement activities where we are focused in our cities communities, and we work with people in London, Poland, Austria and Spain, trying to show our technologies, making walls around our sites, try to design our strategies and so. So to give some achievement, these are some of them, not all. I was a little bit lazy, sorry for that. So we make isolation of microorganisms capable to degrade contaminant. Until now, we also have tested different conditions for stimulation of degradation with the different organic amendment. We designed DNA constructors for a specific metabolic pathway. We have the installation of wetland, the installation of biopiles. We remove contaminants from Baltic Sea. We have a lab, mesocosmos scale test for muscle ability to remediate contamination, and also we have several active questionnaires, discussion and events all around Europe. That is for now. Thank you for your attention, and this is SYMBIOREM.
Thank you, Ruiz. Now we move to the next project, that is EDAPHOS, and will be presented by Michel Chalot from UMLP.
This one, right?
Yes. Thank you very much. So EDAPHOS is a 5-year project granted in a soil mission call. We have 12 partners involved, and EDAPHOS is about land rehabilitation. So we work at 7 case studies: contaminated agricultural soil, brownfield. We have also mining sites, and the idea is to set up some plantation that I will detail a little bit later. And we want also to improve biodiversity using this nature-based solution. We want also to improve ecosystem services, to restore ecosystem services, and for that we have a number of indicators that are going to be measured during the period of plantation. EDAPHOS is built around 3 technical pillars, I would say. There are 2 other pillars that I will not detail today because they have not started yet. Pillar 1 is about mapping the pollution with some remote sensing technology. So the idea is to map the pollution with some plant indicators. And for that, we are flying over the 7 case studies with drones, with aircraft, and with also, we are using also satellite data to have different resolution of information. And the idea is to, again, to map the information that we got from these remote sensing tools with the pollution that is in the soil. And an example here, we have started to evaluate this, the correlation between the plant health indicators and the soil contamination, and some of the indicators are indeed useful for that. The second pillar is about evaluating the risk in the different case studies associated to the pollution, not only before but also after the implementation of the nature-based solution. So for that we applied the so-called triad approach, which is made of chemical indicators that we are measuring at all case studies. We are here a table with all the metals, for instance, and you can see that some of the sites are contaminated with cadmium, copper, etc. The triad approach is also made of ecological indicators, which allow us to evaluate the biology of the soil. And you can see that here we are measuring enzyme related to carbon, nitrogen, phosphorus, or sulfur cycle. And you can see here, for instance, at the Spanish site here, everything is in red. So meaning that this soil is very poor and there's not so much vegetation growing there. The third indicators that we are measuring concern ecotoxicology. And for that, we are a partner that is involved in measuring a number of indicators. And we are also involving a company, a small company, which is called Microfluidics, who also apply this type of tools as an innovation to the project for evaluating some of the toxic effect of the pollutant in the soil. And you can see again here that we have here some red colors for some of the sites, for some of the indicators, meaning that also this site is quite toxic for a number of organisms. The third pillar is about the implementation of nature-based solution. So for that, we are working with a company who is providing us with new poplar hybrids, because poplar is a very good species, a relevant species for producing biomass on contaminated soils. And here, for instance, we are comparing the poplar production on different sites, the poplar production with different hybrids, and we are measuring production of biomass and also accumulation of trace element. And the idea is to select the best hybrid and to co-crop this hybrid with, and this is one of the innovations in the project, to co-crop this poplar, these trees, with herbaceous species to improve the exploration of the soil and to improve the captation of pollutants. So we are working with different plant species that we are setting up at the different case studies. Also, we are improving the growth of the plant by using microbes, and this is linked to the BIOSYSMO project, because we are using the resources that we are producing in BIOSYSMO to improve the growth of our plant species at the different case studies. So, in EDAPHOS, we are trying to link soil contamination with plant health indicators. We are trying to restore biodiversity, and for that we are measuring the efficiency of nature-based solution that we are implementing. And the idea is to work with both combination of plants and combination of plant and microbes. And here is the list of partners. We are 12 partners in this project. I thought I had another one, but no. Okay, so that's it. So if you want to know more about EDAPHOS, you can go to the website or scan the QR code here. Thank you very much.
Hi everyone, so I'm here to present pHYBi, a very new addition to the ALPHA-BIOREM cluster. This project mostly focuses on phytoremediation, but not just phytoremediation, but also connecting the derived phytoremediated crops into high-value bio-based product chain. So this project is going to last 4 years. It started very recently and last year in October, and it'll go on until September 2028. It has a total budget of 5 million euros and 4 different case studies. And this is a collection of the different partners. I think many of these partners overlap with the other consortiums that we have within the ALPHA-BIOREM projects. And now I will go on to explain a few of the objectives of pHYBi. So this project workflow is slightly different to the one that we have seen in this cluster. It starts with the investigation and monitoring of existing phytomanagement trials, but also looking for new candidate sites where phytomanagement practices can be applied. And the idea is to basically learn from our findings. So like there's so many projects within this cluster that use these strategies. So we want to use the lessons learned in all of these projects in order to improve the ongoing phytomanagement trials. But not just that, it also focuses on the extraction and the valorization of the lignocellulosic biomass from the phytoremediated crops in order to convert that into coatings and dyes for textile applications. And then, in addition to that, because we just don't do phytoremediation, we also do modeling, we have the development of an open source virtual replication tool, which will basically model all these different components of the project in order to identify which is the area that requires optimization. And in addition to that, like in other projects, we'll also have evaluation of the technical, economic, and social viability of the project, and then have targeted activities for social and stakeholder engagement. The workflow is very similar, and I'm sure most of you are familiar with this. It starts with looking at site investigation and characterization of the different feedstocks. In the second half of the project, we focus mostly on the phytomanagement strategies in the field and also the collection of data. But in parallel to that, we also have feedstock valorization and extraction of the biomass that is retrieved from existing phytomanagement trials. And then once all of that is done, we go on to sustainability assessment and modeling and replication, so the establishment of the virtual replication tool. And then the other work packages are traditional, like the ones in other projects. Going a little bit more deeper into the case studies, we have 4 different case studies across Spain, France, and Croatia. And the objective, like I said before, is to focus on 2 existing case studies, which are former agricultural cropland, which has been heavily polluted by raw domestic wastewater, which is contaminated with lead, copper, zinc, and cadmium, and also polyaromatic hydrocarbons. But then we have another case study in which which is a post-industrial brownfield, also contaminated with heavy metals and hydrocarbons and polyaromatic hydrocarbons. In the category of new case studies, we have some salt-affected agricultural soil, which have high salinity, so require a very special microbial intervention. And we'll be looking at some phytoremediation strategies pertaining to that. And then we also have land which is polluted by organic contaminants, and this is also an area of intervention where pHYBi will establish some new phytomanagement strategies. In terms of the innovation in the project, we'll be focusing on the utilization of symbiotic inoculants, which is endophytic fungi, plant growth promoting bacteria, and mycorrhizae in order to boost plant growth. The more plant growth, the more biomass, and the more the high value based product. And then we'll also have a nano-bio-phytoremediation approach in which we'll basically be using iron and carbon-based nanoparticles in order to, in combination with bioaugmentation with native microorganisms. This word is too big for me to pronounce it, so I'm going to use native microorganisms in order to enhance tree-assisted phytoextraction. And then, in addition to that, to support the implementation of these strategies, we also have genomic scale metabolic models, which will basically help us take a deeper look into how are the plants and microbes interacting in order for the plant to be healthy and happy. So, in parallel to this, we have feedstock valorization. Now that we have the biomass from these phytoremediated crops, we will basically implement an organosol fractionation in order to derive the hemicellulose, lignin, and cellulose fractions. And then these will be then directed into another workflow where they will be used to generate nanocellulosic-based coatings. And this can be applied. One of the industries would be textile and fashion industries, but of course it can be also have other applications. So with all of this, I just want to highlight that pHYBi's objective is a little bit unique in the sense that it has 2 different objectives. So it's the restoration of contaminated land, but also redirecting the biomass obtained into another value chain where you generate bio-based products. And both these strategies will take place in parallel. So in one line, so in the first line, we see that we will focus mostly on the biological aspect where we will monitor, apply, and implement phytomanagement trial sites. But then apart from that, we also have another line in parallel where we are basically using we are collecting the biomass obtained from these polluted lands, and we are redirecting it in another value chain to generate products that could be used in another in the industry. So in order to summarize, pHYBi helps clean polluted soil. It also gives the plants higher resiliency against environmental stressors, but it also helps us transform these lignocellulosic biomass from the phytoremediated crops into nanocellulosic coatings and C5 sugar-based pigments. The advantage of this is basically that we create a closed-looped value chain in which we are not just cleaning up the soil, but we are also connecting that with biomass valorization and generating high-value-based products out of it. And this aligns very well with the EU's goal with circularity and the synthesis of a low-carbon process. We also have some fancy technologies in the project, which is remote sensing, soil monitoring, our online virtual replication tool, genomic scale metabolic models, which will also increase the transference of innovative phytomanagement strategies within pHYBi to hopefully other projects. And then we will also have a lot of activities targeted towards engaging with industry stakeholders and helping people accept these products better in the market. Thank you.
So now we move back to Michel to present the project IASIS.
Okay. Yes. Thank you. Thank you again. So this presentation has been prepared by the coordinator, Effie. I'm not sure it fit within the time slot allocated, but we'll see. So IASIS is the sister project of pHYBi, the same call. We have 17 EU partners in this project, 1 industry, 6 SMEs, and a farmers advisory SME as well, and academics, of course. Well, I go to this one. So the IASIS is organized around 4 technical work package. One is about the optimization of crop production at the site. So we are working with both contaminated and saline sites. The second work package is to set up field trials at different case studies. The third work package is about the development of added value by product and products. And the fourth work package is about evaluating the sustainability and the feasibility of the whole value chain. So the key elements of IASIS is that we are going to grow a number of industrial crops at the different sites, improving this crop production by using microbes, you can see here. So we have designed a number of consortiums, so we are isolating microbes from the different sites to build this consortia, or using existing consortia. And then the idea is to apply these solution to a real field situation. So we have 8 case studies with saline soil, and we have 8 case studies with contaminated soil, metal contaminated soil, mostly, and pH as well. And the idea is to produce from these crops a number of byproducts that I will detail in the next slide. So we have these 8 different plant species, cranberry, cannabis, etc. Some of them are shared between different sites within Europe. So the sites are located in Portugal, Spain, Greece, France, Belgium, and Poland. So here we are cropping plants from the different contaminated soils. And here the idea is to produce, to process those plants, to extract a number of useful metabolite or useful product, which are listed here, some intermediate product, but also some end product, and to use them for bioherbicide application, for instance, or as coating materials. So here we have a number of partners that are involved in the processing of this biomass. And here it's about the lignocellulosic crop. So we are growing a number of plants such as nettle, hemp, salicornia, to extract cellulose, hemicellulose, or to extract other molecules. And the idea is to process those molecules to produce a number of end products such as bioplastics, biocomposites, etc. And again, there are a number of partners here that are involved in the processing of these different molecule and byproduct. So the impact is, of course, to increase availability of feedstock and to work on, let's say, contaminated saline soils will allow to bring an additional soil surface to produce these crops. We want also to improve, by development of guidelines, recommendation, to improve the communication with the principal stakeholders. And we have a whole set of farmers that are involved in the project. We have 10 farmers per site, so we have a total of 80 farmers as stakeholders that are involved in the project. So it's a whole community that is also working with us. We will train them for using a number of tools that we are developing in the project. And the idea is, at the end of the project, to decrease the contamination or the salt stress at the different site by applying this practice and the production of byproduct that goes together with the harvesting of the crops. So here are the different partners, 17 partners from Europe. Thank you very much.
Thank you. The next project is Life MySoil, and it will be presented by Geoffrey Herrero from Eurecat, soon Barcelona.
Thank you. I will try to stick to the 5 minutes. At least I will try it. Life MySoil project, the difference. So it's a live program, different from all the other projects that you hear. The main difference is that it's co-financed, so it's 55 budget from the European Union, 45 from the companies or entities that are part of the consortium. And the other main difference is also because we start from the TRL 5.6 and we have to reach 7.8. So it's mainly, we want to have a final result, a technology that the companies can already have in their to exploit, to give it to their clients. So Life MySoil project, another difference between the other projects, just ended this month. It has been a almost 4-year project. It ended we did the final event on final of May. And today I will try just to show you 4 slides of how the project has been. Mainly we focus on petroleum hydrocarbon pollution in the soils. It's one of the most frequent pollution. There are several techniques, and within a life project you have to have a baseline strategy and compare with the technology that you want to develop and fit into the market. So it's not just efficiency, it's also LCA, LCC, socioeconomic, and so on. And it's mainly about microremediation with bioaugmentation with lignocellulosic fungi, the white rat fungi, that I think we already talked about it at some of other sessions. Moving on, what we did, and most of the other bioremediation strategies for the soil have to do. First, they focus on the site characterization. In our case, we had 5 pilots plus 8 replicability sites. Then we performed some biodegradability tests, from the adaptability test to the microcosm, half liter to 5 liters. So from half liter to 5. Then we moved to the small pilots, also we call it mesocosm experiments, most of them already in the field, so they are, the meteorologic conditions there has been affected. And then from the small pilot, we moved to the pilot micropiles that has been a 50 cubic meter, with a total of 3 sites performing this technology at this scale. Okay, MySoil technology. So mainly we developed 2 different strategies with the static micropiles or static biopiles, mainly because if you want to grow fungi with hives, if you do a dynamic one, they will break. And with these static micropiles, we have tested 2 different approaches. One, the usage of spent mushroom substrate, mainly Pleurotus ostreatus, Agaricus bisporus, but also with the isolation and bioaugmentation of fungi in 2 other pilots. And also, since we have a pilot where the pollutant was fungicide, we couldn't apply a living mushroom, so we moved to the enzymatic extraction from the spent mushroom substrate in order to treat it. And in that case, we mixed the bacterial bioaugmentation plus the enzymatic extract. Regarding the monitoring, that I think we didn't have a lot of time to explain that in the last Congress. We mainly develop a multi-line evidence approach, apart from the conventional one. We moved to the GCMS screening, where we check, since you know that the TPH is a huge cloud of pollutants, we should divide and see which parts are more recalcitrant and see if our fungi are able to degrade compared to the bacteria one. Also, we developed, or ISODetect, a company part of the consortium, developed a mycotrap that is a in situ microcosm with a labeled 13 carbon and deuterium, in order to see that really the fungi and the autochthonous microbiome are degrading and incorporating this carbon. Moving on, the PLFA, just to check which are the communities from the fungal and bacterial part that are degrading the pollutant. Of course, molecular genetical tools, a lot. All the other projects of the cluster are also applying. We also perform toxicity tests from the soil and from the leachate. And finally, I presented yesterday the guidelines. The last slide, main results. I tried to give an overview. The micropile, the pilot micropile, we had between 40 to 60% of removal. This removal, it doesn't seem a lot. We reach a plateau of 1,000 ppm of TPH, but we wanted to be transparent. So the initial pollution was between 6,000 to 10,000, but between you excavate, accumulate, do all the experiments, start the pilot, it was already between 2,000 and 3,000. Therefore, it was the more recalcitrant part that was there, and we were able to degrade that part. What else? The inoculated fungi, they were really active the first weeks. In the middle of the pilot, they were decreasing, and at the end of all the treatment, they were not even detected by molecular techniques. We had a very good results compared with the baseline. Our baseline was thermal desorption, about 90 to 99% of reduction of energy consumption, and 80 to 99 reduction of global warming, but we also detected specific points that could decrease this reduction, since the transport of the excavated soil and the transport also of SMS from a long distance. Okay, moving on, specifically the microremediation was better compared to the classic biopile, for the kinetics and for the specific or more complex TPHs. But at the end, both strategies have a similar reduction at the end of the treatment. And some points that maybe we have time to talk about in the panel. The main obstacles or barriers that we found that are common with other projects, the site-specific biotreatability and microbial adaptation. This takes time before applying to the pilot. Space and time constraints. There are not a lot of places that they allowed us to have an excavated site during 6 months. Then we move to the soil treatment plants and infrastructure, which are very diverse within the different countries of Europe and even the different regions. Bureaucratic and regulatory constraints between regions, lack of standardized methodologies and risk assessments for TPH. So TPH as a whole, it's quite tricky. We'll publish in our website a white paper regarding this topic. And yeah, the criteria of how the sustainable is the technology should be included and when you choose a strategy. Finally, after Life MySoil, since in 15 days will be over, from the research part, we'd like to focus more in the decrease of this recalcitrant fraction, try to decrease this plateau, move to the degradability of other pollutants. We tried TPH, HTF, PCBs, but there are others to try. I would like to decrease this time and human resources and money of the preliminary test and according with the cluster, with these predictive molecular tools and digital twins and so on. Regarding the business applicability, they are working mainly with the brownfields and refineries, which they have availability to have piles there, but also with other soil treatment plants. Thank you all, and that's it.
Thank you, Geoffrey. I don't know if somebody is going to present the BIOREM project on behalf of Rocio.
Okay, you're ready.
Yes, Feliz, thank you. So sorry, Rocio cannot attend today to this presentation of the BIOREM, so I will present for this. First of all, I want to say thank you for the organizing committee for inviting us to this interesting cluster. This is actually the first time that we officially present this project, so we are excited for this. BIOREM projects became at the end of the last year as a part of the Eurozone Europe, especially the Marie Curie actions, and that the main objective of the Marie Curie action is the exchange of the research staff and interdisciplinary change of the different knowledges. So this is the reason why in our project we have different partners of different institutions. In the case of partners of companies, we have a Kepler, and also we have Limnos in Slovenia that are a very expertise in the bioremediation process application. And in the case of the university, we have the University of Surrey and Wageningen, and also the Joseph Stephan Institute as a research center. So why BIOREM? So the main objective of BIOREM is or was born by the necessity of to find new solutions due to the growing presence of the contaminants into our ecosystem and soils due to the human activity. And in these contaminants, we can include persistent organic pollutants, solvents, metals, TPHs, PAHs, and pesticides also. So the framework that BIOREM project proposed is the bioremediation of contaminated soils with targeting in pHs and PAHs, but also in combination with other contaminants like methyls and PCBs. For a first step, a solid characterization of soils will be performed to identify which are the possible processes that are including in the bioremediation, and also a consortia isolation, a microbial consortia isolation will be performed to understand all this process. This microbial isolation will be identified, and omics tools will be integrated by metagenomic, metatranscriptomic, and metabolomic analysis to identify the different microbial networks that are in this that are performed for the bioremediation process. Bioinformatics also will perform this process by the identification of the different genetics expression, the metabolic pathways, and among others, to assessment the functional and understand what is the functional of the different consortia that we can isolate in these soils. So all this information will be employed for the application of different strategies in this contaminated soil by the use of different biostimulation strategies and microbial-assisted process like biostimulation, smart carriers, synthetic bacterial community, or microbial-assisted phytoremediation. The main proposal, the final proposal of this experimental situation is that we can include the artificial intelligence and machine learning to increase or to create a predictive models that can be applied for many of these situations and contamination in different soils. So the objectives of the BIOREM project will be coordinate all this information and all these new ideas for increase the bioremediation and biodegradation process in this type of soils. The BIOREM project will be also integrated different disciplines like soils, omics, and artificial intelligence to understand these degradation networks. In this Marie Curie action, we have mentioned that one of the main objectives is that all the partners can change information and scientific data for search and innovation solution in this field. So all this staff, all the research staff will have secondment in this sense and will be lead one of these main keys according to the Marie Curie actions. This is physical and analytical chemistry and computer science that will be led by University of Burgos. Also the applied science, that the more applicability part that is performed with Kepler and Limnos, and finally the genetics, genomics, and bioinformatics that will be used by the University of Surrey and Wageningen. So finally, the impact expected in the BIOREM project is to enhance the career or the perspective of the staff members by the collaboration between the academy and the industry. It's also to establish a long-lasting collaboration networks with for the future works in the field of the bioremediation also, and the knowledge transfer between the different partners to create new ideas in the field of the bioremediation process, and also to research and innovation potential to field of the in situ bioremediation of organic pollutant and mixed contaminant. So thanks all, and this is the information. If you want to contact with us for any question, we are available, so thank you.
Thank you. So now we will move to the panel discussion number 1 that will be chaired by Fabio.
Yes, and we start moving.
Yes.
Thank you very much. Maybe I sit in there. Oh no, we need another seat. Do you have a seat? You do not have a seat? No, it's impossible. 2, 8, 9. Indeed, we miss. Well, I can stay here, okay? So thank you very much. First of all, let me thank Philippe, Professor Corvini, for this idea to bring the running projects in some ways together. This is very, very important. Very important because you have the chance to share also technical issues related to how to implement the project, the interaction with the Commission and so on and so on. But in particular, you can share your experience, your know-how that you are developing. And this is very, very relevant always, always relevant. And the other point is, please, this is something that I'm asking, but I think is an important point. Also share your views and put these views in documents, short documents, position papers. Surely Elisabetta Balzi, Dr. Balzi this morning, but this afternoon too, mentioned what is coming. But I can tell you that the Commission is also listening, is very open to listen what is coming from those who are implementing innovation on the frontier in the sector. It's a good moment for bioremediation. Back, it was a good moment in FP7. Now there are interest in bioremediation again, and in particular in the restoration also of ecosystems and biodiversity. So not only removing pollutants, but also in taking care to the health of soil, health of ecosystems, because they can only be productive in such a way. As you know, the health of environment is connected to our health, the health of animals, plants. So one health point on which also the European Commission is now working a lot. Also combining 5 different agencies of the Commission, they are working together for setting up a new Sheen under One Health. So be active also in working for producing position paper, short, effective, coming from your experience and also from the discussion of this afternoon. Bioeconomy strategy is open, still up to 23rd. So bioeconomy is also looking at inputs from the environmental biotechnology part, for many reasons, again, the ones mentioned and others. So please do also this. Philippe maybe is in some places, but I think it's very important that you can also go in this direction. And you can do this if you are together. Yeah? Okay, thank you very much for what you did and what I'm sure you will do soon. And, well, we have some topics on the table. You know the topics, right? Because probably you identify jointly. The idea is to ask you a few statements. You are many. There are 4 points on the table. Surely 2 are quite relevant. All 4 are relevant, but both 2 surely interesting. Also for the reason that I mentioned it to you earlier. One is the the emerging technologies in the bioremediation. What is your feeling about the emerging techs? This morning we work on the electrochemistry associated with, it's not a recent one, but interesting, producing nowadays more data than some years ago. And the other point, the enabling role of microbiomes, the microbial systems, and also how we can stimulate, if you have the way to stimulate or to... These are 2 points on which I think we can start, and then there are other 2 points, as you know, right? You know you have four points to address, but I would start with these 2. So new tech, among them I would ask you at the same time to refer about the role of microbiome. Of course, if microbiome is at the center of the sheen of your project, otherwise you can have general feedback. I don't know, I guess that I didn't study enough, I didn't share enough, so maybe we go with the microphone, right? One of you are referring on this, okay? Short, because we have little time. No, you go for, okay, thank you. Can we record the talking? No. You are recording too.
Yeah, you can hear me, I think so.
Okay, great. Thank you. No, just for listening again. Thank you. So please, go ahead.
Okay, related to your question, I think the most interesting that you mentioned before is that the microbes are the main targeted problem, or not problem, the main issues that we can now study in this relation to the biodegradation of the different contaminants in all of the statements, because all the relation that we have in all the system, all the strength that we can include in all the things that we can see in this process. Omics tools, I think, is the most revolution recently in this context, because you can get a lot of information about genetics, about the relation of the microorganisms, and you can improve this relation or this function of the consortia or the microbiomes for increase also the biodegradation process. So I think this is the most targeted issues that we can go ahead with. We can investigate more in this field, because I'm sure that we can get a lot of information for improve the bioremediation process.
Thank you.
Okay, so actually similar.
Be closer. I think it's green. No, it's working. You simply have to keep it closer to you.
Okay,
closer to you. Thank you.
Following the idea, I mean, we cannot deny the interest of the microbiome, how is, let's say, the story, how everything works in the early minimum stage, a gene, but also we have not to lose the idea that this should work. I mean, should work in full scale. We need to reach up our objective, that is try to have as clean environment as we can. So this is where the multidisciplinary have good challenge also. Biologists speaking with technologists, or technician, or engineering, or chemists, try to find the help of electrochemistry and put everything together with a final result of having more clean environment. It's super good, it's super valuable to have clear idea that why and how the things are happening, but we have to go also further, make it happen in a full scale.
Thank you, Leire. I totally agree with Leire regarding the full scale. Indeed, Life My Soil focus on the TRL 7.8 to really prove a technology. One topic that I think my colleagues will talk better will be about modeling digital twin of the microbiome. I think it's the revolution this year's and the next years. I'm not an expert, and I think it's a need for most of the projects. Some topic that I think during this conference we saw, it's the synergy between different strategies. One strategy alone won't be able to remediate, at least at full scale. It will be integration of two or more technologies or a sequential of technologies. All bioremediation or maybe some bio and the other physical or chemical one. And I have other things like, I didn't find where to put it, but we should work more with companies that would like to go to the field scale. I didn't know where to put this idea, but we really need more experience and more companies trying and willing to do innovation in bioremediation.
Thank you.
So my expertise lies in genomic scale metabolic models, right? And that has taught me one key aspect of microbiomes, microbiology. In terms of pathways, in terms of capacities and resilience and adaptation, they're far superior to us. They persist in environments that are harsh. They are extremely complex. They can take carbon dioxide and convert it into a multi-carbon compound. So keeping all that in mind, that's why we're putting microbes at the core of our technologies, right? And that's why we're studying microbes. So this is why microbiome and microbial communities, microbial consortium, all of them play a very important role in our technologies. And this ultimately connects to the other topics that we will discuss in terms of what technologies do we use to understand them better? How do we improve and scale their performance in broader bioremediation application and field scale applications? So how do we exploit this extremely advanced regulatory capacities of microbes in bioremediation and really actually use it as a nature-based solution? So I think it's important that we really understand the capacity of the microbes and make an effort into understanding it better, and that's how we can better scale it to address, I don't know, remediations at different scales, at different TRLs, at different capacities of the project.
Thank you. Thank you.
Yeah, I totally agree with Aki, because I'm from a company, and we do soil and groundwater remediation. We also do lab feasibility tests, and we had some projects where it worked perfectly fine in the lab, but when we went to the piloting or did large-scale remediation project, we couldn't replicate what we've seen in the lab. So there are some knowledge gaps. So usually we don't really check for the molecular biology. We work like more or less with black boxes, and this is a huge problem. So when we see it's not working, we need to investigate why it's not working and how we can improve it. And this is like a thing we really need to target to get a better understanding of the pathways and how the microbes work together, or which key factors are missing. So why remediation is not really working. And just one point, it's really great that everyone is, or like the community is starting to focus more on these microbiomes, because I think this is a huge benefit. Because if you have a single strain and he doesn't like your environment, he will just simply die. So, and usually we target like on sites several different contaminants and not only a single one. So you have a broader range of degradation pathways when you're working with a microbiome. So the success is usually better with microbiomes.
Thank you.
My colleagues already mentioned a lot of issues, and I would like to kind of bring back something that was already mentioned in one of the sessions in this conference, and it's about data sharing, and not only data sharing but also data standardization, which in the end, to understand the microbiomes and to understand the technologies and why they work or why they not. We are a lot of projects doing a lot of work, and sometimes there is no these mechanisms to share data between us. Probably for the omics there is more work already there done to share data regarding omics, but I think that we should go a step further and to think how we can standardize the data from other type of experimentals, from lab setups and from pilot setups, because then if we have that data available, we can use models, and we can use artificial intelligence to exploit that data, and maybe we can find some answers to those questions.
Thank you.
Yes, actually I fully support your last point. Of course, microbiomes are in nature, so we should find a way to exploit them in the best way. The issue is that, let's say, of course, every situation, we know every site, every situation is very different. We are generating a lot of data because each project individually is producing many omics data, and also the metadata that are associated to these projects, to these tests. And yes, probably the way to better leverage the potential of the microbiome exploitation is trying to put everything together and, let's say, also obtain information that we don't know where it is. Somewhere it is there in the web, in the databases, and probably with the use of artificial intelligence try to, let's say, combine and find real information in this large number of data that we have already for sure produced, and that we are going to increase much more.
Thank you.
Yes, I fully agree with you and with what has been said before. Omics, modeling, artificial intelligence are innovative solutions that can be easily applied to microbiomes. We have seen also this week some innovative approach, such as the microfluidics. We had the chance this week to have a company representing microfluidic here. We have seen application during this week within BIOSYSMO project, within the EDAPHOS. In BIOSYSMO, for instance, we construct some microbial consortia using microfluidics. In EDAPHOS, we are developing ecotoxicological tests for using microfluidics, so that can be readily applied on soil. So this is an innovation. But on the top of that, I think also some progress should be made on how to apply the microbes to the soil, because this is also an important question, not to work only at lab scale, but also at real situation, real environment scale. How we bring the microbes to the soil, how we bring back them to the soil. There we have seen some kind of some solution, how to encapsulate them, etc. This is important because when we apply microbes, we don't know if they're still alive after 6 months, 1 year. So it's important to be able to detect them and to protect them once they are in the soil. And the other point I would like to emphasize is also it's important to involve the end users, because if we build consortia, microbial consortia in our lab, but if we are not able to, if the end users are not able to use them, then we fail. So it's important to train, and that's why what I mentioned in IASIS, we are a whole bunch of farmers that we will train for the capacity to use the crops, but not only the crops, also the microbes. We want them to be able to use the microbes that we are producing in the different labs.
Wow, thank you very much. Well, the points has been clear, and I can also only comment a couple of things. One of data sharing. This came out also maybe yesterday or the other day, I don't remember, is crucial anytime and for any sector, but surely here we should use more, in particular infrastructures, because it's not easy to store data, also because we need to check the quality of data. We need to store and then make them available up to a certain point, because they should be used in a proper way, not misused or improperly used. This is a priority. European Commission, again, is taking care to this, research security, but we need to, this is one statement that you should put in one document if you wish to go for. The engagement of companies for sure is a very important issue, and also the final inputs on technology, biomentation, microbes immobilized. The bioelectrochemical approaches that were presented also today are now improved. These are other statements that we can place, you should place in a document. And then also the other, you mentioned many things. Microbiomes are surely strategic, but we should be able to manage, to in some ways push them in the proper or in the right directions, and so on and so on. And so, Philippe, are you coming? Are you joining me now? Because I also need to leave. I came late and then I leave to go to the taxi to go to the airport. So the other 2 points were, you know, right? The other 2 points, you distilled the questions. The other 2 points is how the bioeconomy, I can comment only and then I leave the floor to Philippe. The other 2 points, one is both are interesting. The one is dealing with the possible synergies, mutual benefits in between bioremediation and bio-based industry. Bio-based industry is an emerging area in the large chemical, pharma sector, etc., in which we are making bio-based products for different applications. There are prominent tools that the European Commission launched for boosting it, but there are connections in between bio-based industry, bio-based products, and your role. Yesterday, somebody proposed polydrosyl-canoate for stimulating in situ bioremediation because they are slowly degrading substrates that can support the microbes in charge, for example, for reductive dechlorination and so on. So this is another point that you agreed to discuss, and then Philippe will manage the discussion. The other point, again, is the valorization of byproducts coming from environmental remediation, because you are also producing byproducts that has to be managed. And nowadays we have to be circular, so in some ways we need to, take care to them, but with the idea to reuse them for internal benefits. So these are other final reflections that can be of huge interest for all of you. I leave the floor to, and then I'm moving gradually to the taxi. No, I left the 2 questions, the 2 final 2 questions. They are, you can go through and
okay, comment on that? Okay. I think regarding the link with the bioeconomy, we have 2, maybe more examples of projects that are linking the microbiome with the bioindustry, is IASIS and PHYBi. We are improving crop production on contaminated site, on salty soil, in both IASIS and PHYBi, maybe in other projects as well. So the link is clear. We have the microbes, we improve the crop production, so we improve the production of byproduct and end products that are useful for the industry. The only thing is that we are working with marginal lands, contaminated lands. Then it's important also to follow the fate of the pollutant, because they will end up, especially when it comes to metal contaminated site, the metal will end up in somewhere during the processing of the biomass, so we have to take care of that, and to be sure that treating this fraction where the pollutant ends up will not cost too much, otherwise economically it will not be sustainable. So it's very important to follow the fate and to evaluate the cost associated to the treatment of the contaminated fraction. We have experience with that. We have, for instance, tested some poplar biomass that were contaminated within bioethanol production, and we know that the contamination is ended up in known fraction, and then the cost associated to treat this fraction is not negligible. So we really need to evaluate all the process to be sure that it's cost effective.
Yes, I have regarding this link between bio-based industries and bioremediation, for sure, there is some opportunities in some cases to valorize byproducts coming from the bioremediation actions, and I think this is probably the case when you have a plant or some biomass that is accumulating a pollutant, either organic or metal, etc. But of course this might be somehow more limited situations, I don't know. Of course we should also exploit the opposite exchange, so maybe find a way to valorize byproducts from the bio-based industries, or from, let's say, in bioremediation efforts, so that anyway a mutual beneficial interaction can still put in place. Of course, if the bioremediation target on an organic pollutant is mineralization, then let's say, hopefully, I mean, the aim of bioremediation is to leave no byproducts in terms of pollutants, but we might also try the opposite direction in order to make this link, linking bioremediation with bio-based industry.
Okay, so regarding the valorization of these byproducts, since I still have a lot of colleagues to probably talk about that, I would also like to sort of make the statement that in the end we should also remind that apart from possible byproducts, we have two main products, which is clean water, or at least treated water, and treated soil, which I think that sometimes we forget to put economic value onto that, and we are not, since we don't put that economic value, we are not able to communicate well that economic value. But for example, if you have a wasted land that is polluted, you cannot develop any urban activities on this land, so it's really economic loss. And I think that this, since the degradation of soil and the pollution of water and the water scarcity is going to increase in the future, I think that this is something that we should all always remind to emphasize, because I think that there is already some inherent value there for the bioremediation.
Yeah, thanks. I will try to answer a bit for to the other question. So, how soil remediation can also help the bio-based industries. So I can talk for my company. We also try to look after byproducts or waste products to use in remediation projects and also for cultivation of bacteria actually. So I know from university usually go for mineral salt medium or high standard substances and chemicals, but it is definitely worth to look if you can find some alternative nutrient sources, which are usually declared as waste. And especially in the food industry, there are many sources you can use to cultivate your bacteria or to use as a nutrient source directly in the remediation, and this helps both sides because the food industry, it's a waste product, and for the remediation you can save a lot of money when you take when you use these sources, and I can just encourage others to look also to these products because they're worth it.
Thanks, Jessica. So I think this question kind of pertains a lot to the objective that we cover within the pHYBi project, where we're actually targeting not just the phytoremediation, but also the valorization of the contaminated biomass obtained after phytoremediation into and redirecting that into the production of a bio-based product. However, I would like to also emphasize that here we are talking about bioremediation technologies, and I think we have reached a certain level of innovation when it comes to the demonstration of bioremediation technologies. But I think the same level of commitment and perhaps innovation is required in order to redirect this all the products that come out from cleaning technologies into this bio-based value chain, especially because whenever we have, in the case of pHYBi, we have probably biomass containing higher quantities of metals and pollutants. So we will ensure that these byproducts will be treated efficiently in order to get rid of all these carryover contaminants, and also that communication is done in a way that the people, like the society, who's ultimately going to be the end users of these products, are aware of the fact that even though, despite of the fact that it comes out of a bioremediation setup, does not necessarily mean that it's not usable. So I think we have to technically enhance the extraction of the full value of the biomass, but also communicate to the people using this that there is a possibility that you can use materials that are generated from bioremediation technologies and that they're not harmful. And science and technology will ensure that all of that is extracted and safely stored, as Michelle pointed out, so as to not have any byproducts that are left after the bioremediation is done. So I think I'll just conclude this by saying that it's not just the technology in terms of bioremediation, but enhanced technology in terms of full utilization of the waste, of the generated waste products, and also effective communication with the people in the society for them to use it.
Thank you. I think pHYBi project is the perfect example to answer this topic. Just two small comments before passing the mic. In MySoil project, for instance, we reuse the agricultural residues. That's okay, but not comparable of what the other projects can do. And one specific comment that it was one of the take-home messages that we realized after MySoil. Doing the LCA is really important because even the same technology, the same approach in different zones, they could have a really different impact. I mean, from cradle to grave, we realized that sending by truck or moving some biomass that it's not locally produced from another region will have a huge impact and will decrease a lot the benefits of using one bioremediation or other. And also the concept, I'm not an expert, but this is safe and sustainable by design. It's something that we should apply or iterate during the technology when we scale up and we are replicating it. That's it.
I will add end of life. So safe and sustainable by design and end of life. It happens to me that the half of my research goes to remediation activities, the other half to bio-based materials. So I am super happy to have these reflections. I mean, there are a huge advance in material science, a lot of money put for the European community on this area, and somehow, even if I am not involved, pHYBi or the project under pHYBi call meet together two very valuable ideas, how we can bioremediate, how we have this also phytoremediation. From that we have lignocellulosic residue. Lignocellulosic, they can be put together for making resins. With that resins we can go back and make a very nice polymer to help a bioremediation process, and once and again do it again. For us, in SYMBIOREM, for example, we are using, by the way, secondary streams. It's not nice to say that those are waste streams. Are secondary streams that we need to use. We use fruit residue to enhance our bioremediation technology. And by the way, they work very fine, very good. We have residue, secondary streams, put it as nutrients for our microbiota, and works good. I mean, let's do it. And take for all the residue of phytoremediation. We have sativa that is very nice, hemp. From hemp, for ages in Europe, we have made many textiles, and it's valuable resources.
Yeah, I'm not to oppose some things more than my colleague says, but I think that the relation between industry and or bio-based industry and the academy is clear. When we apply a bioremediation process, we obtain it different bioproduct, or obviously we need to research more about this type of bioproducts. But for example, there are many microbiomes that, or many bacteria, for example, that can accumulate metals, that can recover metals for soil. So this is a value for bio-based industry, and create a competitiveness and increase the value of these methods of these products for the industry. So I think we need to increase this change of information in the bio-based industry and the academy for increase this competitiveness. The academy needs to help to the industry to increase their productivity, to increase the use or the production of different products or different value products for increase their process and their activity.
So now it's time to change to the second part, and I will not go the question in the original order. So there's been a subject of discussion on how involving other colleagues, and notably those from the blue technology, biotechnology, and listening at the talk of Elisabetta Balzi, I think it is time now to make some thought on how to have a kind of unification and attract those persons. And this is the case for ocean, but my opinion is also when we talk about bioremediation, and so we talk about natural attenuation, and then the monitoring, and this is something that is also mentioned in the forthcoming topic. So I would like to hear about your opinion here.
Okay. In the case of Philip said about the blue synergy, and in the case of the topic of the marine ecology, I think that we have the same contaminants in all the topics in soils, in water. All we share pharmaceutical or petroleum hydrocarbons, for example, when we have an oil spill. So in this case, I think that all the person that we are working in the different topics, bioremediation in soil, in water, in the different area, we need to share all this information to increase the share, the data for the bioremediation of these contaminants. So yes, it's necessary to increase the transfer of the information between all of the colleagues. Nowadays, with the exactly contamination that we have, that is this soil or water. So yeah, I think this is the main topic that we need to focus our studies.
Yes, I mean the key aspect I believe that we have to focus is, we have already speak about, is how to share between different science, different scenarios. We can bioremediate in soil, we can bioremediate in oceans, we can obtain materials from the oceans, like polysaccharide or active molecules, and go and use them in bioremediation of soils and water. So this exchange of technologies, this exchange of material, this exchange of resources or knowledge should be more fluent between the different areas. We are super happy to go and be in our lab, but we have to be forced ourselves to go out and speak with colleagues working in microalgae. Microalgae can help us with the bioremediation. You don't need to know about microalgae. You have to find a good friend and make science. That is the point. Have good friends. Don't be shy to ask for help. Improve your network, and the science is going to be great.
Okay, I'm the third already, and it's hard already to add something new. So totally agree, there is a multidisciplinarity within the bioremediation of soil and groundwater and surface water. If we go to the ocean, of course, totally agree that you don't need to know all the technologies, all the scopes, everything. There are some techniques or methods that are shared already between the different fields. For instance, the modelization with the genomics, of course, it's quite similar. The pathways will be different, but the pathways of the degradations, I mean, but there is a common floor. Here, maybe my question or my reflexión, my introspection, perfect, thank you about that, will be that, okay, this cluster, it's good, we are doing dissemination, we are doing panels, we have shared goals, but at some point, maybe we have to grow more, or we have to be another, not a cluster anymore, but a association or something like that. But the cluster will evolve, will change the objective, and this change should be made by all the coordinators and the projects and the partners in agreement with the new goals or the new goals that we are planning.
Thank you. So I'm just going to steal the answers of my colleagues, but maybe say it in a more technical way. I think it's important that whenever we are working on these projects and we are generating so much data, we think of these, not to sound commercial, but we have to think of these as products. Products that can be taken to other projects. Products that have to be applied or implemented in field scale applications. For example, I see the microbial consortium as a product that could be taken to other projects, that could be manipulated and put in new context, like from terrestrial to marine. But I think the point here is that in order to do so, we have to understand the technical implications behind it. So we need to know if this data, monitoring data, for instance, that is being collected in a project is being done in a way that it's actually going to benefit connecting with the projects that will be carried out in the future. So we may, I mean, the European Soil Commission, or the European Soil Mission has made so much emphasis on how the data should be collected. There's so many standard laws and regulations about it. I think it's very important that old projects and new projects collect the data according to those regulations so that it ultimately, that monitored data can be used in modeling strategies to make useful interpretations, but then also these modeling tools that have been based on correct data can be applied in other future projects. So I think we need to think about this in more technical details and how they can be actually, what are the actual steps of connectivity between these projects and what are the products from these projects that will be transferred from one to another. So, yeah.
So for me, there's not really much to add. It's a bit tricky. No, I totally agree. So I think the most important thing is really to connect also with other projects. I think we have a great example between BIOSYSMO and MIBIREM. So there's site materials sent between these partners shared, their data shared, and this is how you can really proceed in science. And when you are open, when you share your data, when you build like consortia, like the ALL4BIOREM consortium, so when you come together, as we are doing here, then you can proceed and get new ideas and transfer like from soil or to ocean. I mean, these are two completely different materials, but there are some connections, and but when you brainstorm together, you will find solutions. So this, like, yeah, coming together and sharing data is really important.
This is very much going in the direction of EIC Pathfinder portfolio.
Yeah, so well, I would like just to elaborate on one comment from Jofre, that regarding the reach of the cluster, which what I think is that if we want to increase the reach of the cluster to topics, for example, the ocean mission, I think that we already did that. We have the example with the soil mission, that we included some projects into the cluster, and we also have the example of the CBE, that now we have also 2 projects from the CBE. And what I think that how we reached that, how we achieved that in the end was to leveraging the expertise of interdisciplinary partners that we already have in the projects. So we already have also partners in the projects that are focus on blue economy. So, for example, my colleague Ana Paula, she just made a presentation about their data platform that is fully focused on the blue economy. So I think that we can just repeat it again. We had partners very focused on soil, so then we reach out to soil projects, soil mission projects, and we have already partners very related with the ocean mission, who probably already have projects in the ocean mission, so we can use their expertise to know how to reach out to this kind of partners and projects.
Yes, of course I'm fully supporting this expansion of the cluster also to the oceans, looking at bioremediation. If I can comment, I have a feeling that the ocean, I mean, we can apply the same approach. Basically, they may be working and adapted. My feeling is that, however, on the ocean, maybe from a technological point of view, we are a bit, let's say, behind compared to terrestrial systems in terms of deploying technologies for remediation. I've been working or studying the biodegradation of pollutants in marine sediments, and always struggling then when thinking about the possible deployment of the solution in the real site. So I think that if we aim at enlarging this cluster also to the ocean, we should somehow think on how to involve projects that are also developing technologies in this direction, because maybe projects on the oceans are still a lot on the monitoring, on the modeling, maybe not. I don't know about all the projects, but maybe a bit behind on the development of technologies to deploy the solutions for bioremediation. I don't know how we can do it, but we should pay attention to this aspect.
The other thing I would like to mention is that there are already lots of cluster existing. In EDAPHOS, we are part of 6 clusters. We should take care not to add another level of cluster, because we will end up with spending all of our time in clustering activities. I think it's already a lot, so we need to think how to organize these clusters and how to make sure that we're not spending all our time. Today we are 8-10 different projects, so half a day it's okay, but if we are more projects, more clusters, then we will need 1 day, 2 days just to talk about clustering activities. And then it's going to be complicated to have, as you did this week, to have half a day on clustering activities, which is very nice. But having 1 day, 2 days on cluster activity, it's going to be too much. So we have to be sure that we are not overwhelmed with cluster activities. And maybe we could also a little bit discuss about the other. There is a mission on global change, climate change, which is also linked to what we are doing on terrestrial, on the management of soils, for instance. So there are clear links with that also that we could highlight within the next few years in new projects. We are already addressing this aspect with carbon sequestration, for instance, in soils. But I think there are maybe more things to do with linking the soil mission, for instance, with the climate change mission.
So this brings me to the next point, that is the link to the health and One Health strategy. How do you see our contribution in the frame of this strategy, and how could we maybe more prominent or visible in that area? Because I remember the 2023 BIOREM with the opening of Larry Wackett, and he had an issue that was directly a health concerning issue with the chlorinated compound in the swimming pool.
Yeah, maybe here the link is easy. In the soil directive, for instance, there are a number of indicators that have been described that are useful. We are all measuring these indicators. Some of them, not all, but most of them are linked to the soil characterization, and some indicators also relate to the biodiversity. But in this directive, for the moment, there's nothing linked to the human health. So maybe we could introduce a few indicators in this directive. We could think about adding these indicators. There are a number of indicators related to human health that are available, that are measured by a number of scientific groups in Europe. So maybe we should think about adding these indicators of soil health. In EDAPHOS, we are measuring some of them. They are usually more costly than just measuring soil indicators, but I think it's worth using these health indicators for the soil, related to the soil, and to how the solution that we implement to our sites can improve soil health. And for that we need indicators, so maybe that's the way to go, having more health indicators, human health indicators.
Yes, I agree on the human health indicators, but we should find a way. This is again a story that is always a site-specific, and actually, one thing is the soil health. A different thing will be anyway the impact on the human health, on the contaminants present in a site. I think in order to relate better, typically we do risk assessment to understand if the exposure of humans to the contaminants is posing some risk or not. So probably this is a way to push more on, let's say, on the risk assessment when we develop and test new technologies, because maybe some indicators may be relevant for certain sites or certain situations, but may not be, let's say, relevant in general, and probably you need to find something that is site-specific, or you can do site specifically for that site.
But risk assessment is based on indicators.
Yes, sure.
To be able to measure something.
Yeah, sure. Yes. But risk assessment in terms of exposure is based, of course, on indicators that we already have. I don't know if...
But they are not in the directive of the soil.
Okay, yes, not in the directive. Okay.
We have one external.
Sorry, I just want to tell you something more about the soil directive, because my colleagues at the JRC, they are now dealing with the watch list for the soil directive. So the idea is to have a monitoring program for soil, listing the most concerned substances, and using the same approach that we use for the watch list in surface water, where we look at the substances that may concern not only for aquatic organisms, but as well for human health, including, for example, antimicrobial resistance. It is important really to clarify this aspect, that you are talking about indicator for human health, but now my colleagues are working on soil in this respect. I take the chance, I don't know if you mind, to tell a few words about this cluster, what I heard today. And I think you have really many common stuff that you should look more and work more together, because I saw that in some cases there are field studies, few, while could be complemented by others. Companies, I didn't really hear too much about companies, but if we want really to increase the competitiveness, this one should be more, let's say, more active. And other aspects, you talk already about integration, data, metagenomics. Maybe Giulio knows, we work on metagenomics since years. There are data. The data are there. It's just to put together. And the other aspect, which I am more favor, is the modulation of microorganisms that are already in the environment. Thank you.
Thank you, Teresa. Yes?
Okay. Okay, so yeah, just to follow up with, I do agree with Giulio about the need of site-specific risk assessment. But what I think is that I think that the Commission and probably the JRC has can really help a lot, because what we find when we want to apply these methodologies is that then each country may have different requirements or different guidelines. So in the end, it's very difficult to select what methodology is the best one that we should apply. And in the end, what would be the best is that we try to be consistent and apply homogeneous methodologies so they are comparable, and then we come back to this data sharing, and then the data that we generate is also comparable between us. So I think that is a two-way that probably we have to put our necessities, as Fabio said. Maybe we have to put it into paper and to see, okay, we see these barriers that we are facing, and then from the Commission they can work from the top to address those kind of barriers.
Yeah, thank you. So I can speak up on behalf of a company. Yeah, I think we're really, or I'm in the minority here. So I think it would be a great thing to have a human health risk assessment, but I need to say that this must be also cost efficient, because what we see as a company, when we do an offer for some problem owners, they always try to reduce the price, they always try to reduce the amount of analytical costs, the parameters, so they always need to reduce it. So this thing would be like, it would be necessary that it's cost efficient, and best thing would be that it's really like mandatory, so you couldn't get around it, so you would need to do this and couldn't skip it. And this is like something concerning like the authorities. So the politics would need to change laws that this is like really a thing you should do when you're assessing a new site, and not like an option you could do by your own, because then like extra cost won't be covered. Because, yeah, site owners, problem owners, they always try to save money, and every additional cost is a point of discussion. So, yeah, costs are a main thing we deal with as a remediation company.
Yeah. What we do at the JRC for water, where I can see the water directive is more advanced than the soil. That's clear. But for soil directive is also coming. And for water, we also look at the risk assessment for the complexity of chemical pollutants in water, implementing the biological-based methods. And nowadays, in the review of the Water Framework Directive, for estrogenicity, there is now mandatory, and we are already doing interlaboratory exercise for testing the validation, across several co-workers, and we are doing the same for mutagenicity. So this kind of bioassay, for example, could be also implemented during the bioremediation phase to check and monitor eventually products that are released, degraded, and could also lead to some effects. So this one could be something that we could think about. Yeah.
So thank you so much for making that point, actually, because that is more aligned with how I would have responded to this question, because here we are talking about bioremediation, we're talking about innovation and so many different types of technologies, but I think we can take advantage of the same innovation and the same technologies in order to think a little bit more beyond what we are cleaning up, and think about if there are any harmful intermediates, if there are any compound levels that need to be reassessed, if there's any gap in terms of how we understand the contaminant concentration at a certain site. So basically using scientific technology to re-evaluate what is toxic, how is it toxic, are we actually truly cleaning it up? So I think that would be one of the biggest contributions that we can have as innovative project owners towards One Health. the reassessment of everything, the reevaluation of everything. And then also one of the obvious objectives that we have is we are all working with bioremediation projects. So I think one of the obvious benefits that we have is we are actually truly cleaning up the soil or water. Yeah, so we can just continue doing that. But yeah.
Thank you. Several things. I'm really interested in this soil watch list. Indeed, I was following during my master time and PhD everything about the water. Really interesting how will you focus this watch list in soil. Regarding the human health risk assessment, it has been a topic that within the consortium of MySoil, we discuss a lot, because depending on the country, the regional level, the human health risk assessment is a mandatory step for the companies to do. For instance, in Spain, each region has its own method and maybe different database for the toxicology, with some drawbacks because it's not an accumulative toxicity, it's just for a group of pollutants. Other countries, for instance, France, they don't have thresholds, so we also talk about the law and the regulations and which is the concentration that it's safe. France, for each big site, will define due to the very detailed risk assessment, but doesn't have the law with the different use of soil that other countries have. On the other hand, we go to Italy and you see that, you are above, of what is that pollutant, but you cannot refine by a human health risk assessment because maybe in that soil use there is no risk, but you have a law that you cannot sobrepasar or surpass, overcome, exceed. Perfect. Sorry about that. So on one hand, human health risk assessment is something that the companies are already doing. Maybe they can go another tier or a more detailed. Something that I was thinking from the beginning, the part of the ecosystem risk assessment, it's something that needs a little bit more research or applied research in order to do it correctly. And also, I move back to the SBD, the Safe and Sustainable by Design, that my Technological Center, EURECAT, already applied to several projects, so they are gathering the human health risk assessment plus the LCC, LCA, and socioeconomic. So they are merging everything together, plus also the environmental risk assessment. It's something that it's there, it's already in the European calls, but and some companies are not the most innovating companies are already doing. So the seed is there. We need to water in the right direction.
Yeah, no, I agree with you, but we need to hear it. I already hear so much. I mean, it's really to move forward. We need to do something.
I somehow linked to that. I mean, for me the picture is I have the chance to get a grant. I develop very super fast and super nice and good working technologies. I'll go to the field. Maybe the technology development take me 1-2 years. If I am lucky, I get the permissions. I go to the field. I stay there like 1-2 years at most, and the project finishes. So the monitoring is stop. And this, for me, is a huge problem. I believe that somehow there should be a tool that allows us, even if we are super lucky, could be ourselves continue working in the same area and so, but also the connection to other calls in which are doing monitoring of the environment to actually know how the performance was in midterm, because the risk assessment are super good, but are theoretical. There is no somebody making analysis, looking how the ecosystem is performance, how the environment of the neighbors, the health is going. So we mix, or at least from my point of view, I mix some tool, some symbiosis between projects to allow us to make this follow-up, not in 3 years project, something that mix together and continue the project, because I mean the soil has their own times, their own direction. It's not 4 years because we say so. So for me it's a huge problem on that.
Yeah. In my cases, I totally agree with the Michelle appreciation to include the indicators for the health risk in our project, because all of us usually apply a technology for the bioremediation. We see that what is the concentration of the contaminant in soil or in other matrix. We apply a technology and we see that this is the depletion of the contaminant. But which is the real fate of the contaminant? Who is the biodegradation of this contaminant and the risk associated to this different substance? Because in many of the cases this contaminant is not totally degraded, it's transformed in other substance that can be toxic, not only for the organism, only for the humans. So it's necessary to integrate this type of risk assessment to see if our bioremediation is okay, or is completely environmental friendly, you know. So yes, another point of view that I wanted to, or other appreciation that I wanted to point is that maybe the public health can be integrated in this kind of topic, you know. The expert in the public health can support to the scientific community in this topic about which are the risks that we are see in this type of contaminants. So yeah, I think this is the question that we need to resolve in this case.
Okay, so we need to change the battery. We'll go to the next point, and I think it's great. We have colleagues from the European Commission, and it's about sustainable financing of activities, and also, as mentioned, the lack of resources to go beyond monitoring what has been done. I think there should be some tools like I don't know, CSA or even cost action that could fit to our purpose.
Okay. Yeah, I'm not very expert in this topic. I think I know that all my colleagues, they are more experienced in all these things. But yeah, I think that all the projects that we are see in the last year, like I don't know, Project Life or other ones, needs to include a more integrated of all the young people, all the interests of the different community to need to our interest in this topic, in the bioremediation, in the biodegradation. So yes, I think we need to involve all the people in all the discussions for this topic, and this is all the things that I can.
Yeah, I mean, it's not easy to solve, to create one scenario where the goals or the project can link together cross sectional or however it was to connect the project. I mean, this cluster could be an idea, but this cluster is covered by the project that we are involved. So it's a kind of something that could work for a moment, but it could be regulated how to continue the work of the legacy of the previous project, continue the monitoring, and try to don't lose good technologies, and also how those technologies that already goes to the field have performed. Is very challenging, is not easy to solve, but it's a problem that we have right now, and somehow and somewhere or someone should face it.
Okay, I would like to say 2 or 3 small points. One, that it's not only Horizon, there is LIFE projects, there is Prima projects, there are several that most of them are, you can develop or scale up the TRL of several technologies. Also, during this Congress, we found out really nice projects, maybe nationally based, that could be part of this cluster or another cluster from a national way. So there are a lot of projects going on. What else I would like to say? Sorry about that. Yes, the stakeholder mapping, or having clear where are the companies, but where is the public administration? Because most of these projects, I assume that the public administration, from the environmental point of view, they are doing a following up of the projects going around the region or the country, and might be the ones even more demanding that sometimes the European Union for the national partners. So I think that we need this kind of stakeholder, the public administration, putting more pressure on us and avoid repeating what we did before, doing other stuff.
Yeah, so I think the financing of the different projects has been really conscious of the gaps that have been identified in the ongoing projects. I think there is a very good correlation between ongoing projects, the gaps identified, and then the call for new topics always targeting and addressing those gaps. But maybe it would be nice if there's some communication from the side of, like, from the identification of the innovative technologies that are being discovered or implemented in the ongoing projects, and in order to exploit that even more and taking that forward through collaborations with other projects, through this cluster, for instance, and that could be one opportunity of using the finance, right? So if you have currently worked on developing an innovative system, you can take advantage of being in a cluster, partnering with people that you were probably not in your consortium, and then taking advantage of that technology into a new call. So I think that would be one of the ways that we can use the financing opportunities. And the other strategy would be, so it would be nice to have some assistance from the European Commission in order to identify these innovative technologies, the kind of direction to guide a little bit the new project writers on which direction they should take in terms of applying for finances. This looks promising and this does not. Just that classification would be really helpful, but some more open communication.
Yeah, so it's always hard to be in the middle because most points have been mentioned. Yeah, so what I would like to add is also that what we said also in minutes ago, it's important to share data. So not everyone is starting at the same point again. So is funded and starts to do the same thing maybe another group has already done. So like the same work is paid twice. So data sharing is really important. But I think it's also great to include companies into calls like this and also try to valorize even small parts out of these research projects. I mean, it's nice to collect data. It's nice to publish paper. This is all nice. Also field tests are nice. But we really, as you said also before, we really need to do something. We really need to go into the field and do full-scale remediation projects. And therefore it's important, even to fund small companies and really to go out and use the knowledge and not just do more and more research, but use what you already have.
So just to add, Well, for the financial sustainability of projects, I completely agree with Joffre about other calls. In terms of the cluster itself, yes, Philippe mentioned probably CSAs or cost actions could be explored from a cluster point of view. But I think that I understand Michel's concern about growing and expanding the cluster, but I think that is something that we really need to do to then be in a better position to search for this financial stability, because I think it would be a better selling point if our reach is larger than it is today. And yeah, I think that there are other clusters that have that were born from this type of activities, and now they are kind of self-sustained. But I think it was because of that, because they included many projects and even projects from different calls, and not only Horizon and companies for sure. So I think that that's the way we should continue.
So I must say, I don't know how this could happen, let's say, to use available EU funding options to extend our activities, even in particular beyond the projects. I think the only possibility is to win another one and establish the bioremediation cluster with the new sister projects. Probably. So maybe this kind of let's say clustering activities, of course, suggested by the topics, could be maybe something that might become mandatory, maybe with the suggestion or let's say already indication of which topics also should cluster in order to let's say put together the efforts and harmonize the efforts. But I think that let's say having I mean, the cluster will be changing in composition over time, because some projects will end up and the new ones will start. So probably to make this cluster alive over time, we should accept, of course, that is going to change composition in terms of projects, in terms of involved institutions, but let's say maybe institutionalized in terms of let's say presence, topics that in each program are already pre-grouped, maybe in the call, and not only invited, let's say, but somehow obliged to participate to the cluster activities, maybe also with some budget in the call, in the topic dedicated specifically to this activity, and maybe also some common deliverables. I don't know. I mean, this could probably or could maybe a way to keep this clustering activities moving on in the next year, and not maybe ending up with this group of projects.
This is excellent, because we will launch the new soil pollution cluster next week. On Tuesday afternoon there will be an online meeting for this new cluster, which GRC is part of. And this is interesting what you just said, because in this cluster we have been discussing a lot on the topics that should be prioritized. And among them, we think that what you just said, that producing common deliverable is something that is very interesting. So we have listed all the deliverable for all the projects that are part of this cluster. We have a list of all deliverable, the deadline also, because it's important to be on the same agenda. And we are thinking about submitting common deliverable for different projects. So this is a very good initiative that we try to develop in this soil pollution cluster. It's very interesting. And also in this cluster, we address the gaps that Aki was mentioning, but others as well. Prioritization of substance, for instance, is something that we want to discuss in this cluster, and maybe come up with a position paper on this prioritization aspect, because in some projects they are addressing this aspect. For instance, in ISLANDR, Aragon, which are the sister project of EDAPHOS, they are addressing these questions. So we'll come up with a position paper on that, but also on the selection of indicators, which are the best indicators to select, which are the best solution to apply, etc. These are the topics that we will develop will address in this cluster of soil pollution.
So this is a nice transition, because the time for this session is over, and I wanted to invite Agnieszka to the stage, because you all received a preliminary draft of a position paper related to the circular bioeconomy. And for those in the attendance that would be interested in commenting on that document, there is time until Monday. Monday. So if you are interested, so please contact Agnieszka.
Yes, absolutely. This is what Fabio Fava just said, because now we have the open consultation for new bioeconomy strategy, and it's the public consultation are open till Monday, till 23rd of June. So I prepared some position paper. It's more or less generic, so it's I think nothing controversial. It was sent, I think, to most of you. Thank you very much already to some I think project which gave me feedback. So still, I think Friday, Saturday, I think it's the latest, because we have to compile it, yes, in one document. And of course, later on, I can send it directly to some people which are responsible for this bioeconomy strategy, but it's something really huge and big at the moment at European Commission. I think you can admit it, it's some important topic. And the next, I think, round, which we can do, it's a Circular Economy Act. It will be the next fall, so it's 2026. So we already can think about, again, to really keep going and show the bioremediation as important aspect of bioeconomy, but as well of the circular economy. And I think to go out of this bubble which we are here and to show a little bit to the world.
Thank you, Agnieszka. And now, maybe Mr. the technician, if you could switch the beamer. Simona has a very important announcement to share. No, come on. Yes, well, I think.
So first of all, thanks to all of you, and especially to this section of the ALL4BIOREM clusters that has been quite illuminating on the perspective that we have in front of you, and yeah, I mean, spill over in different European mission and whatever. So the important announcement is that we will have the BIOREMED conference in 2026 in Florence, and it will host the next ALL4BIOREM clusters. And yeah, I think we hope to have more people joining the clusters, because today, in a way, we are a sort of a selected community, even though they were in the frame of another conference, the European Bioremediation Conferences. So we are also open to suggestions that already came from some of us to organize a more dynamic or more inclusive meeting of the whole 4 BIOREM clusters. And yes, it will be hosted in Florence, so you won't have the opportunity to go to the seasides, but you won't be even the opportunity to go to visit art, because that week will be very hot, so you will be very happy to be in a air-conditioned environment, and we will discuss about bioremediation for all the day. Yeah, I mean, and we will open museums only during the night. We will ask to the council to open museums only during the night. And so you are welcome to join us in Florence next year. It will be in the weeks between 22 and 26, and the cluster will be exactly on the 24. And we are thinking to dedicate all the day to the clusters, so to the presentation of the results, of the further results of all these projects, and perhaps we will have other projects that will join the clusters, and then the discussions, and the following day we will have the BIOREMED conferences with all the delegates from also other fields. And we hope also to be able to invite to join the clusters also the ones that are working on blue economy, on whatever will be an improvement of our bioremediation activity also in other fields. So we wait for you in Florence next year. Thank you.